Okay, this video is a video to help you get through the Hardy-Weinberg introduction worksheet. Before I show you step by step how to get through the math, I want to talk about the data sets 1 and 2 that are in the BCR prompt, which I will come back to at the end of the video again. But I want to talk about the idea of what is an allele frequency. So an allele frequency is the percentage of each allele in the gene pool. So if you look at data set from year 0, the initial data set, the dominant allele frequency is 0.5. That means 50% of the alleles in the entire population are dominant, and that would mean that the other allele frequency would also have to be 0.5. If you add the allele frequencies together, they have to equal 1. So 0.5 plus 0.5 is 1. And inside the Punnett square are genotypic frequencies. This is the percentage of each genotype in the population. If you add all 4 of these genotypic frequencies, well, it's actually 3 genotypic frequencies. I'll explain that in a second. But if you add all 4 of your decimals inside this Punnett square, it should also add up to 1. So in this square, in this top left square, is the genotypic frequency of the homozygous dominant genotype. Bottom right square is the homozygous recessive genotype. And in these 2 squares are the heterozygous genotypic frequencies. So if you want to know what the chances of getting a homozygous recessive genotypic frequency are, if you have the allele frequencies, it's 0.5 times 0.5, or 0.5 squared, which is 0.25. So if you have the TI-84 plus calculator, your squared button is the x squared. And so if we wanted to do 0.5 squared, we would do this x squared button and hit enter. And it's 0.25. And you probably can't see that, but the button that we're using is this one. It is diagonal and up to the left of the 7. It's right above the log button. You're going to be using this button a lot. Now, if we were going to work backwards and we wanted to do, if we had the homozygous recessive genotypic frequency, so if we knew that 25% of the population had whatever this homozygous recessive phenotype is there, we could figure out the allele frequency, because if the genotypic frequency is 0.5 squared, if we have the genotypic frequency, the allele frequency is 0.25, but we need the square root of that. So if you have this kind of calculator and you need to do the square root, you would hit second function and then the x squared, and then you put 0.25 and then you hit enter, and it would give you, I don't know if you can see this, 0.5. But anyway, that is the button, this button that you're going to use. So if you need to square something, it's just the x square button. If you need to square root, it's second. So it's the second function, and then the x squared button, and then you just type in the number that you want to take the square root of. So now I want to go back to I'm going to scroll to the top of this file, and I've made this, much bigger so that it's easier for you to see in a video. But in this, we would be taking the number, so number 1 asks you to calculate the allele frequency of the dominant allele. So to do that, you count the number of dominant alleles and divide by the total of alleles. So if I'm counting this, we have 2, 4, 6, 8, 10, 12, 14, 16, 18, 20, 22 total alleles. Then you count how many of them are big T's, because that's our dominant allele, and divide by 22. And that is going to be our dominant allele frequency. To get the recessive allele frequency for number 2, we just count the number of recessive alleles and divide by the total number of alleles, which is also 22. Now, when you're filling in your data table below, your yellow dominant alleles, which I highlighted in yellow, go here, and your recessive allele frequencies go in the pink. So we put our dominant allele frequencies in the yellow and our recessive allele frequencies in the pink. And these 2 numbers, the numbers by each little t will be the same, and the numbers by each big T will be the same. Now, Basically, what I'm going to talk you through is the instructions that are in this data table. So this data table tells you the instructions for exactly how to fill in your Punnett square. So basically, this video is me reading through the instructions for you. So once you have the dominant allele frequency and the recessive allele frequency, you can figure out all of your genotypic frequencies. In order to find this square, big T, you take this dominant allele frequency and square it. It's the same as taking the dominant allele frequency times the dominant allele frequency here. These two numbers will be the same. So it will be whatever that number is squared. So you use that X squared button on your calculator, this guy right here, or you can just, if you had a older calculator, just take the two decimals and multiply them by themselves. Now, to get your homozygous recessive genotypic frequency, you just do the same thing, but be careful, you need to use the allele frequency from here and here. Since they're the same, you could just take whatever this allele frequency is and square it, or multiply it by itself. To get your heterozygous genotypic frequency, it is your dominant allele frequency times your recessive allele frequency. And then whatever that equals here, you can just copy and paste from here to here, because you're going to do the same thing over here. You're going to do your dominant allele frequency times your recessive allele frequency. Now I've color coded that. If you're wondering, well, how do I know my dominant and recessive allele frequencies? I told you how to calculate them here. So whenever you get your yellow answer, your yellow allele frequencies go here and here. And when you get your pink allele frequencies, which is your recessive allele frequencies, from number 2, from right here, you will just place those allele frequencies here and here. And then you're just multiplying them together. And the reason this works is because if you want to know the chances of inheriting two alleles together, it's the product of their individual chances. So product means multiply. So you're just multiplying the decimals here. Now, this is the hardest part. Once you have this part, the rest of this is much easier. So one thing that I noticed in the few that I've graded already is a lot of people just skipped number 4. It says copy and paste your math from your Punnett square for your genotypic frequencies of procrastinator's disease. And it says, look in the data table for your homozygous recessive genotypic frequency. It is in the bottom right-hand corner. So all you have to do for number 4 is go to here and copy and paste whatever is in this square and paste it here. Now, number 5 says how many people would be carriers of the disease. So carriers can pass it down but don't have the disease. So there are heterozygous individuals. So what you have to do is add the two decimals from your big T, little t squares in your Punnett square. So whatever your decimal is here is going to be the same as your decimal is here. You add those together, and then you multiply by the total population, which in the problem is given to you. We tell you it's a population of 700. So always show your work. Show the numbers that you're multiplying so that your teacher can figure out what you've done. In case you screw something up and mess it up, you want to know the teacher to be able to figure out what happened. So now here says how many people would be phenotypically normal. Add your homozygous dominant genotypic frequency to your heterozygous genotypic frequency, and remember that there are two heterozygous squares, and then multiply by the total population, which is 700. So you go back up to your Punnett square, get your decimal from here, plus your decimal from here, plus your decimal from here, add them all together, and then multiply that by 700. Now, that is for number 6. For number 7, how many people would be procrastinators? You get your homozygous recessive genotypic frequency, which is down here in your little t square, and you multiply that number by your total population. So that is one type of problem where you're given the allele frequencies and you're figuring out the genotypic frequencies based on the allele frequencies. But in reality, it'd be very unlikely for you guys to know, or for anyone to know, the actual allele frequency first. What's much easier to actually observe is the number of people that have each phenotype. So we're going to do a problem where we have brown-eyed people and blue-eyed people. And on this island, and we also should know from the genetics unit that blue eyes is recessive and brown eyes is dominant. So we have 50 blue-eyed people and 800 brown-eyed people. And number 8 says the first thing to do is to figure out the total population. So you add these two numbers together and that's your total population. Now, those 50 people that have blue eyes have the genotype little b, little b. Those 800 people who have brown eyes, well, some of them are going to be big B, and some of them are going to be little b, and we can figure out how many would be big B, and how many would be big B, little b by doing our Punnett square. But this is the key information here. If we can figure out the phenotypic frequency of blue-eyed people, we can also figure out the genotypic frequency of the homozygous recessive genotype. So we would take 50 divided by 850, and that answer goes here. So we're dividing the number of blue-eyed people, which was 50, by the total population, which was 850. And so our answer to number 9 goes here, and it's going to be a decimal. Now that decimal goes right into here. Exactly. You don't have to do anything to that decimal. Whatever your answer is from 9 goes in the square that's labeled 10 in your Punnett square. Don't do anything to it. So then it says in the instructions above, to get number 11, to get the recessive allele frequency, you take the square root of this answer. So you have a number here, it will be a decimal. You take the square root of that number and then you have little b. Now, this is really the next easy part. So whatever this number is, if you add it to this decimal, it should equal 1. So once you have the allele frequency of the recessive allele, you can really easily figure out the dominant allele frequency because you just subtract 1 minus the number here. And it tells you that here. It says, so if an allele is not recessive, it must be dominant. 1 minus the recessive allele frequency from 11 will be your answer here. So whatever the answer is here, you take 1 minus that, and that's your answer to 12. Then for 13 and 14, you just copy your answers. So your answer from 12 also goes with your answer from 13. They're the same. And your answer to 11 and 14 should be the same. And 11 and 14 are going to be the square root of whatever you got from 10, which you got from question 9. Then the rest of this is really similar to the previous problem. To get the number of homozygous, or not the number of homozygous individuals, but the homozygous genotypic frequency, you would just take your dominant allele frequency times your dominant allele frequency. So this number times this number, which is the same as this number squared, because that's the same thing. And then to get your heterozygous genotypic frequency, you take your dominant allele frequency from 13 and your recessive allele frequency from 11, multiply them together. Once you do that, it's the same thing for 16. It doesn't matter whether you do this square first or this square first, they're going to be the same. So whichever one you do first, you're going to copy and paste the answer into the other one. So 16 and 17 should be identical to each other. Now, it should be important for you to note that if you add 12 and 11 together, you should get 1. If you add 15, 16, 17, and 10 together, you should get 1. Now, that's a good place to stop and check your work before you go on to do 18 through 24, because otherwise they'll be incorrect. Now, to get number 18, you just copy, it's the brown-eyed allele frequency, so you just take your answer from 12 and copy and paste it to 18. And number 9 says, what is your allele frequency for blue-eyed color? It's your answers to 11, so you copy it from 11. Now, for number 20, it says, what is the heterozygous genotypic frequencies? Add your decimals from 16 and 17. So you go up to your Punnett square, your decimal from here, and your decimal from here, add them together, and then that is your heterozygous genotypic frequency. Then it says, what percent of the population is predicted to be homozygous dominant for eye color. So to convert a decimal to a percentage, all you would do is you'd come here and you'd see your decimal and you'd multiply by 100. You shouldn't need a calculator to do that because you probably should know by now that if you multiply anything by 100, you're just moving the decimal place 2 places to the right. So then that's the answer to number 21. Now number 22 says how many people would be homozygous dominant. So take your answer from 15. So your genotypic frequency for your homozygous dominant genotype and multiply that by the total population. And in this problem, it's 850. The last problem, the total population was 700. It's going to vary in different kind of problems what the total population is. 23 asks what percent of the population is predicted to be heterozygous for eye color. So you take your answer to 20 and convert to a percentage by moving the decimal place 2 to the right. And your answer to 20 was your heterozygous genotypic frequency. So basically you would add the numbers here and here together and then move the decimal 2 to the right. So for 24, it says how many people would be heterozygous. So you would take the decimal answer and multiply by the total population. So you can't, whatever the percentage is, you move the decimal 2 to the left back again and then multiply by 850. So now let's talk about the BCR. So I'm going to read the prompt to you. It says a species of moth is found near a remote town. The moth has 2 phenotypes. The dominant allele causes a dark pattern and the recessive allele a lighter pattern. A biologist took a sample of the population, about 1,000 moths, and found that the recessive phenotype occurred 25% of the time, which if we change it to a decimal form is 0.25. All of the other moths, so the other 75%, had the dark dominant phenotype. So 20 years later, the biologist returned to the town. During her absence, the town grew and became more industrial. The biologist noticed that as a result of industry, the area had a layer of soot on it. The soot made surfaces in and around the town darker. The scientists again collected 1,000 moths and analyzed the phenotypes. However, this time the analysis showed that 2% of the moths expressed the recessive phenotype, and that means 98% were dark. So the one thing that lots of my students missed, not lots, but the few that turned it in early, they didn't notice this big bold sentence here that said there are many bird species living in this area who hunt the moths for food. That's going to be really important in perhaps explaining the change in our 2 data sets. So remember we said 25% of the moths, which is 0.25, started out being light colored, and 20 years later only 2%, which is 0.02, had the light phenotype. So what you're supposed to do is explain the changes between year 0 and year 20. Particularly, you should be comparing the allele frequencies. Have the allele frequencies increased or decreased, and why? So if we look at the Hardy-Weinberg principle, there are 5 conditions that must be met for allele frequencies to stay the same. All 5 of these would have to be met in order for the allele frequencies to have stayed 0.5 and 0.5 20 years later. They didn't stay that. So you've got to figure out which of these things you think did not happen. So in order to not change in allele frequency, the populations must be large. There must be no migration into or out of the population. There must be random mating within the population. There must be no mutations, and there must be no natural selection. So in other words, no allele can give the individual an advantage for survival and or reproduction. So the question says, the scientist concludes that the data suggests that Hardy-Weinberg equilibrium has been disrupted. Do you agree or disagree? Compare the early data to the data taken later. Make sure to use numbers from the Punnett squares to support your answer. What happened? Be sure to explain what you think the cause is for the change in the data. So you've got to say which of these 5 things you think, which of these 5 conditions was not met, keeping in mind what we said about the bird species who are hunting and the moths for food. That will probably give you a clue if you take notice of that. And I would say that, one thing that I'm seeing is a lot of students are comparing the numbers, the genotypic and phenotypic frequencies, which are not bad, but I think it's a stronger argument to compare the beginning allele frequencies and the later allele frequencies, and then try and figure out which allele, are the alleles increasing, which allele is increasing, which allele is decreasing, and why. So that is it for the end of my video. That should